So kind of change pace a little bit here. We're going to go ahead and look at some organic compounds in the body and how they pertain to physiology. What I want you to know is examples of these and how they're interrelated to form a functioning system. These are all held together by covalent bonds and are important in the food that we eat, and they also make up our cells. They maintain life and are called organic because each of them contain various arrangements of carbon. So starting off, we'll begin with carbohydrates, which include different types of saccharides. The main monosaccharide that we're going to focus on in this class is glucose, which is used by cells for energy. Disaccharides are two simple sugars bound together, thus they need to be digested so that we can get simple sugars to be used for cellular processes. Polysaccharides are simply long chains of monosaccharides. Glycogen can be found in animal muscle and liver cells, and when blood sugar is too low, the liver will use this glycogen to it will hydrolyze this glycogen or break it down so that we can get glucose into the blood. And when we have too much glucose, it just gets changed back into glycogen and stored in these liver and muscle cells. In plants, they use cellulose for their cell walls and then starch for glucose storage. And we eat those in our food, right? Lipids, or fats, a very diverse group of organic molecules. Triglycerides are kinds of fats that we find in our diets, in like cheese and milk. Phospholipids, shown here, make up cell membranes. Steroids, like cholesterol, are important for hormones like testosterone and estrogen, and bile that help break down dietary fats. Now, not all fats are bad, and we need them for normal cellular processes, but just like everything in life, too much or too little can be harmful. So I don't need you to memorize the aspects of what consists of a phospholipid by any means, but just trying to understand that there are different types of lipids and that they're all important in some manner. Now, proteins. All proteins are made of a chain of amino acids strung together by a peptide bond. In their primary structure, protein function is they're strung together in a peptide bond in the primary structure. Protein function is very much dependent on protein structure, and this is maintained by hydrogen bonds for secondary structure, seen here, and bonding of the secondary structure for tertiary structures. And then multiple tertiary structures can come together to form a quaternary or a fully functional structure. In this form, they have a specific function, but take any aspect of that molecular bonding away through any of these structures, and the protein can lose or change functionality, which has very important implications for downstream effects. They have important functions, such as that of enzymes. All enzymes are proteins, and they aid in metabolic reactions that occur in the body. These enzymes are proteins that act as biological catalysts to speed up chemical reactions. Amylase converts starch into maltose, which is a catabolic reaction, and the opposite, DNA polymerase, builds DNA, and that's an anabolic process. The chemical structure of each must be compatible in order for the enzyme to work. So it's a very specific arrangement for what the enzyme is looking for and what substrate can be attached to it so that it can make a product or change whatever it needs to change so that it can affect whatever it needs to affect or whatever it was programmed to affect by DNA, which we'll kind of get to in a bit. Most enzymes work at body temperature, which is why it's important to maintain a specific range for body temperature so that these enzymes maintain functionality. So looking at nucleic acids now, deoxyribonucleic acid, commonly known as DNA, stores genetic information, and this helps code for different proteins, and it's double-stranded. Ribonucleic acid, or RNA, encodes protein information among other functions and works with DNA to help make those proteins. It's single-stranded. It's consisting of 3 different subunits, a phosphate group, or multiple, pentose sugar, like ribose in the case of RNA, and deoxyribose in the case of DNA, and nitrogenous bases as well. So there's adenine, and cytosine, and thymine, guanine, and uracil, and between the 2 nucleic acid types. So taking a closer look at these nucleic acids, we can see the 3 aspects of them here with a nitrogenous base. a pentose sugar, and a phosphate group. The difference between RNA and DNA can be seen here with a missing OH, so that's why it's deoxyribose. So this is ribose, it has an oxy there, so it's not oxyribose, but it's just ribose. Deoxy, so you took the oxygen away from this one, making it a DNA molecule, or a DNA sugar, I should say. And then we have the different types of nitrogenous bases here. We have 3 different types of pyrimidines and 2 types of purines. We have cytosine and thymine, or uracil in RNA, combining with different purines such as adenine and guanine. So guanine will always pair with cytosine, and adenine will always pair with thymine if it's DNA, or adenine will always pair with uracil if it's RNA. And these nitrogenous bases hydrogen bond to each other based on these groups. And last but not least, we have a very important molecule known as adenosine triphosphate, or ATP. It is the energy currency of the body. It provides energy for other chemical reactions such as anabolism, or to drive cell processes like muscle contraction or neurogenesis. If there's no ATP to use, all energy requiring chemicals or chemical reactions stop. Our cells use glucose to make ATP, which again is the direct fuel for cells, like gas for a car. You can see the 3 aspects of the ATP molecule with the adenine, the ribose sugar, and the phosphate groups. The 3rd phosphate group on ATP is kind of like a coiled spring. Once that's cleaved or taken off, energy, or in the form of heat, is released, and that energy is transferred into whatever molecule is being phosphorylated. So that concludes the notes for chemistry and the last part of this week's material.